Welcome, everyone. Today we will discuss how animals utilize nutrition, digestion, and the different ways of taking food. We look forward to exploring an important and complex topic together. Let's get started. Animals adapt their feeding methods to their size and lifestyle. Chewing, scraping, swallowing, sucking, and brewing are some of the approaches they take. For example, snails chew grass, ants scrape insects, eagles swallow flesh, hummingbirds suck nectar, lice and mosquitoes suck blood, and houseflies brew decaying matter. In the buccal cavity, ingestion of food is the initial process which consists of breaking down into smaller pieces by the teeth. Additionally, saliva are secreted by the salivary glands which help in breaking down starch into sugar molecules, facilitating easy passage through the mouth. The tongue helps to mix the food with saliva and swallow it. The tongue is a remarkable organ with numerous roles in digestion. From detecting the 5 tastes of salty, sweet, bitter, sour, and umami, to tasting, manipulating food, and swallowing, it is clear that the tongue has an important part to play. In addition to this, it also helps break down food particles, making digestion easier. Different regions of the tongue are able to detect different types of tastes, which means we are able to receive a full range of flavors from our food. Having healthy teeth is essential for chewing and grinding food. Humans have a total of 32 teeth, which are divided into 4 types: incisors, canines, premolars and molars. Incisors are for biting and cutting food, canines for piercing and tearing, and premolars and molars for grinding and chewing. In both upper and lower jaws, there are 8 incisors, 4 canines, 10 premolars and 10 molars. A balanced diet and proper oral care can help keep teeth in excellent condition. Esophagus is a tube connecting the mouth to the stomach through the neck and chest. In the stomach, acids and enzymes are released to break down food and kill bacteria. The small intestine is a long, coiled tube and receives secretions from the liver, gallbladder and pancreas. The liver secretes bile stored in the gallbladder to break down fats, whereas the pancreas secretes pancreatic juice to break down proteins and carbohydrates. The intestinal juice further breaks down the food, starch into glucose, fats into fatty acids and glycerol, and proteins into amino acids, and absorbs them into the body. The small intestine is an essential part of the digestive system. Its walls are lined with villi, small finger-like projections, which aid absorption and help increase the surface area of the small intestine, allowing more efficient absorption of nutrients and energy from food. After digestion is complete, absorption occurs, where the absorbed materials are sent to different parts of the body to be used for energy, growth, and other bodily functions, known as assimilation. The undigested food passes on to the large intestine where bacteria act upon it. The large intestine absorbs some water and salts before the waste is sent to the rectum and finally eliminated as feces, referred to as egestion. Ruminants, like cows and buffaloes, have a unique digestive system which allows them to thrive on eating grass. Instead of chewing their food properly before swallowing, they quickly swallow it and store it in the rumen, a stomach-like organ. The food is partially digested in the rumen by bacteria and then brought back up to be chewed again, in a process called rumination. The chewed food then passes into a sac-like structure between the small and large intestine, where the cellulose in the grass is digested with the help of bacteria. This digestive process enables the ruminant to get the most nutrients from the grass, as seen in the image of the 001 buffalo. Amoeba are single-celled organisms found in pond water and other freshwater sources. They have cell membranes, nuclei, and cytoplasm that contain bubble-like vacuoles. Additionally, they possess pseudopodia, or false feet, that allow them to move and capture food. When an amoeba comes close to a food particle, its pseudopodia envelope it and form a food vacuole. This food vacuole protects the particle while enzymes digest it. After digestion, the food is absorbed, and the waste is excreted out of the vacuole. This is the method by which amoeba feed and digest their food. Animals have adapted to different ways of consuming food in order to survive. This table shows various types of animals and the food they eat, as well as the manner in which it is consumed. Snails, for example, chew their way through grass, whereas eagles swallow flesh. Hummingbirds suck the nectar of flowers, while lice and mosquitoes or houseflies have also adapted to their own particular forms of nourishment. Examine your mouth in a mirror and use your index finger to feel the different types of teeth. Count and record the number of incisors, canines, premolars, and molars in the table provided. After collecting the data, analyze it and draw your own conclusions. Learning about the types and number of teeth can help you gain a better understanding of animal digestion. In this activity, we can observe the effect of saliva on starch. Take 2 test tubes. Put some boiled rice in one and some boiled rice chewed for 3 to 5 minutes in the other. Add water to both tubes and then 2 to 3 drops of iodine solution in each tube. The boiled rice will turn blue-black due to starch, while the chewed rice will not turn blue-black because the saliva has converted the starch into sugar. This experiment shows that saliva is essential for digestion. Taste buds are located on various regions of the tongue and are responsible for sensing different tastes. To figure out the different taste regions on the tongue, make separate solutions of sugar, common salt, lemon juice and juice of crushed neem leaves or bitter gourd. Utilize a clean toothpick and place each solution separately on different areas of the tongue. The tip of the tongue is sensitive to sweet substances while the back of the tongue is more responsive to sour and salty substances. Label the areas of the tongue which detect the sweet, salty, sour and bitter substances. This experiment helps you comprehend the various tastes identified by one's taste buds.